So I'm going to talk about understanding immune responses in the Southern Corroboree frog to chytrid fungus. So Sam gave a very nice intro to both the Corroboree frog and the fungus. So I'm cheating a little bit. The Southern Corroboree frog is just outside of Victoria in Kosciuszko National Park, or was. The historic range was in Kosciuszko National Park between 1,300 and 1,760 meters. They are a small ground-dwelling frog, yellow and black, very iconic, very nice looking. And the population started to decline in the 80s, and initially they thought it was related to drought, similar to what they were seeing with the biverns. And then they started to look back later on at historic specimen, and they found that they were seeing BD infection in the 80s. And we think that BD arrived to Australia in the 70s. So BD being the chytrid fungus, Batrachochytrium dendrobatidis. So it arrived in the 70s and then spread around the continent and is thought to have created this decline. So the Southern Corroboree frog in 2005 to 2006, roughly half or more than half of the population in the wild were infected. And then by 2012, they were extirpated from the wild. So yeah, you get a boo for chytrid, boo for extinction. So they're maintained through captive rearing programs at Zoos Victoria, Taronga Zoo, and a number of other facilities. And they're working on trying to get them back into the wild. The issue is there's these common eastern froglets, which are very common in the area that the Corroboree frogs are native to, and are pretty tolerant to BD. And we don't want to boo the frogs because they're just doing what their frogs do, and they just happen to be really good at dealing with this fungus. But they're keeping it on the landscape. So even though the Corroborees have pretty much disappeared from the landscape, the fungus remains. So they're trying to release frogs back into Kosciuszko, but they're being killed by the fungus. So what I want to do for part of my project is look at what's actually going on in the RNA. So who remembers the central dogma of molecular biology? Everybody loves the central dogma. So DNA replication, and then your DNA is transcribed to RNA, and then that RNA becomes protein, which actually creates the function. So what I'm most interested in is what is actually being transcribed. Because not all DNA that you have is actually being transcribed, so it's not actually being expressed. Just because you have the gene doesn't mean it's showing up. So we infected some frogs in the lab, and then we were going to take tissue samples and see what's going on. So we looked at Xenopus frogs, which are a model species, and we wanted to see when they have immune responses. So we see different peaks. So you see early peaks for your early initial response, your innate response, just your body reacting to the invader. And then an adaptive response would happen long term, and it would be a later, more specific, direct to that pathogen. And we also wanted to look at different age classes. So we don't really know much about how the age classes are affected. And this can help inform when we're releasing frogs if there is particular advantages for releasing different frogs of different age classes. So we infected, again, all three age classes, and then we're going to look at how this changes over time. So we've measured our chytrid loads, and then we took tissue samples at 3 days, 7 days, 14 days, and 24 days. So we can really capture both that early innate and then the later adaptive response in the immune system. And we want to compare the differential expression between these individuals, between individuals of different ages, infection status, whether or not they survive, and by the treatment group, so control, so unexposed versus exposed frogs. So we got our transcriptomics data back, and there's 153,316 different genes expressed across the 200 individuals that we got. So there's a lot going on, and we're trying to sort through all that. So first I subset the data by age and day. so we could look at what's going on at each time point. And I'm going to take you just through some of the more interesting stuff, because with that many genes, there's a lot we can get into and a lot of differences. The spoiler is that there is a lot of differential expression, but we don't know necessarily what it means. But we did see that when we compared exposed frogs that came up positive with chytrid to frogs that were exposed but didn't end up getting infected, we did see differential expression. A small number of genes, so we saw 12 genes downregulated and then 4 upregulated in the post-metamorph frogs at 7 days. So this is a heat map kind of showing what that looks like. So each of these columns is an individual, and each row is a different gene. And the colder it gets, the more downregulated it is, and the warmer it gets, the more upregulated it is. And you split them up by groups, so you can see kind of different patterns here. But the interesting thing is these individuals that were exposed and stayed negative kind of cluster together. So they have similar differential regulation in those genes. And we saw a similar pattern with the juvenile frogs, but as you can see, a lot more genes showing up. So 161 upregulated and 311 downregulated. And that's what it looks like when you have that many genes on there. It gets a little crazy, but you could still see this strong pattern. So you could see this line here where it kind of breaks off, but there's a pretty strong pattern of all these individuals showing pretty similar expression. And then at the adults, we saw, interestingly, less genes, so only 2 upregulated and downregulated, if you go to the next one. And the pattern gets a lot weaker, so the clustering isn't as consistent. So you have exposed positive and exposed negative frogs kind of just all over the place. And then we wanted to look at exposed negative frogs compared to the control frogs. So are there differences in frogs that never saw any chytrid exposure to the frogs that never got infected but did? And again, we saw 58 genes upregulated and 57 downregulated. So each one of these blue dots is a significant gene, and the ones that are down negative would be the downregulated, and then your upregulated would be the ones above here. So this is another view of that. So in the juveniles, we saw 58 upregulated, 57 downregulated. And then 106 upregulated and 65 downregulated at day 7. At day 14, this was consistent, so we saw 44 upregulated, 65 down. So we're seeing consistent differences between those exposed and the unexposed frogs, all the way through to day 24. So the number of genes bounce up and down, and we haven't identified all of them and what they are and what they do, but there does seem to be consistent differential expression. And then we wanted to look at, the takeaway from the infection status is that juveniles and post-metamorphs have a pretty similar response. The adults have kind of a similar response, but the patterns are weaker and there's fewer genes. So there seems to be less going on in response to the exposure. By age, again, we saw very similar patterns. I'm not going to show you all the same graphs again because they're all pretty similar. But we saw that the post-metamorphs and juveniles tend to have more genes that are upregulated or downregulated, and the patterns are pretty strong and pretty consistent throughout the days. Whereas the adults, there's less genes and it's a weaker pattern. So the overall conclusion is, yes, there's lots of differential expression. It appears to be stronger in the younger age class. It's hard to say why or what that is. A lot of the genes or the regions that we're finding expression in aren't characterized or aren't annotated, so we don't know what they are or what they could potentially do. But there does appear to maybe be an early innate response, so we are seeing an early response, and then potentially an adaptive response. Don't necessarily know if it's actually adaptive or if we're just seeing some sort of lingering response to the exposure. And we have a lot of genes to examine, so there's a lot still to figure out here. So next steps are to try to identify what those genes are. We're working with, or trying to potentially work with another lab to get more genes annotated figure out what they actually are. We want to run a blast, so look at all the genes that we have, compare them to all the ones that are in a database and see what they do in different things. And then we want to improve that functional annotation so we can get an idea of what's really going on here. But then potentially we could use that to, upregulate or downregulate certain genes that could potentially exhibit some sort of resistance to either to infection or to increased survival. And with that, acknowledgments, so my committee and all the other students that have done so much work and helped us with everything along the way, and all of our partners and funders and our lab.